let's start. Questions पूछिए. हाँ हाँ बोलिए.
You had told us that the curcumin capsules are practically useless. What is the exact reason? Because isn't the essential substance same even if you put it in Indian food preparation or in a capsule?
Yeah, so what happens is that, so basically it is turmeric powder, and the active ingredient which makes it so healthy is curcumin. So the curcumin is extremely healthy for us. Which ways? It is healthy because it is anti-inflammatory. So many of us, raise your hand if you have experienced it. Many of us, when we get hurt, right, we drink hot milk and we mix this turmeric powder and we drink it, right? And inflammation disappears by morning, right? So it is anti-inflammatory. It has antioxidants. So what is the meaning of antioxidants? आप लोगों ने, you must have seen many commercials. ये क्रीम में antioxidants हैं, इस खाने में antioxidants है, fruits have antioxidants, vegetables have antioxidants, right? और जितना the more colorful the vegetable is, the more colorful the fruit is, chances are it has more and more antioxidants. So why we really want antioxidants?
ROS को down करते हैं.
It cleans up ROS. What is ROS? Reactive oxygen species. Why does ROS generates? So the good thing is that, the good thing or the only thing is that we need oxygen for survival, right? All of us need oxygen for survival. The bad part of oxygen is that oxygen can form free radicals, it can form superoxide or H2O2, and they are very dangerous for us. And where do we consume oxygen? During electron transport chain, right? Mainly there. So electron transport chain, those of you who have studied electron transport chain, electrons are going through the complexes, right? So if electrons escape, they react with oxygen, they form superoxide, which is bad. Then superoxide is converted to H2O2 by superoxide dismutase, right? And then H2O2 is converted to water by catalase, by peroxidases, many enzymes, right? So in our body, we have the defense system, antioxidants like SORT, superoxide dismutase, like peroxidase, all of these things. So when we are young, our defense system is very high. So anytime superoxides are formed, they are cleaned up. Anytime these ROS are formed, they are cleaned up. As we age, slowly the defense system goes down. So we start accumulating oxidative stress. So people, as they get older, they have wrinkles. That is due to oxidative stress mainly. So, and that is why older people are more prone to disease. Any disease, more prone. Older people are more prone because they have accumulated too much oxidative stress related stuff in their lifetime. So when you take green grapes versus red grapes are considered even more healthier because they have more polyphenols, right? So that is why more antioxidant, that's why they say vitamin E cream or vitamin C cream, antioxidants, right? So antioxidants are very good for us. So curcumin also has antioxidant properties, okay? So curcumin is very good. Now the biggest problem with curcumin is that it is not bioavailable. What that means is that if you just take 1 spoon of curcumin just directly or via capsules, our body excretes the whole thing. Nothing gets absorbed. When you make it in a curry and you cook it, it becomes somewhat bioavailable. It is definitely bioavailable, but to just a certain extent. So that is used by our body. That's why our inflammation goes down. That's why it is very helpful. Okay, so a lot of companies worldwide we know the structure of curcumin, right? And we are organic chemists. So we can make it more bioavailable. So far, not much success. So now think about it. So the Western world also knows curcumin is very good, but they don't cook the food the way we cook it. So they take the capsules thinking curcumin will help. And I tell them, don't take the capsules. You're wasting your money because it's not going to be absorbed by your body. It's going to be excreted completely. So that is why if you cook it in that way, when you take it with milk, it's always boiling milk. You boil it for some time to make it bioavailable. If you take a capsule and drink milk, it's going to make no difference there. So the correct way of taking it with boiled milk is to actually add it and boil the milk for some time and then take it while it is hot. Okay, so that is why taking capsule is not good, because it is not bioavailable in its raw form.
I had also asked about the angiogenesis you were about to tell me during.
Yeah, so what was the question?
Ma'am, so the question was that.
Speak it loudly so that everyone can hear.
You told us before that if we are treating a cancer by controlling the angiogenesis, it initially works and then it later on does not work.
Oh yeah, I remember that.
So
yeah.
What changes do they cause so that they can adapt to the low oxygen condition if we are?
Yeah, so that is very good question. So what happens is that, so angiogenesis, just to remind you, and please ask me questions because you have very chemistry background, Leknath. So what happens is that Leknath has visited my lab, so we know each other pretty well, right? So, but I'm getting to know all of you. So coming back to angiogenesis, so angiogenesis is formation of blood vessels, right? And so the tumors, they make blood vessels because blood vessels are the one which bring food and oxygen, and they need both oxygen for Krebs cycle. So what happens is this, this is very interesting thing. Let me draw it here, okay? So, so suppose this is the normal tissue here, and the tumor grows here like this, okay? So oxygen and glucose, they diffuse to the tumor. So imagine that this is the blood vessel. So I used to think, if I ask you, if the tumor is growing like this, which will go further, glucose or oxygen? Which will have higher diffusion rate? So raise your hand if you think it is oxygen, and raise your hand if you think it is glucose. So I would raise with you guys, but all of us are wrong. Glucose goes further than oxygen, and I was very surprised to see that because I thought it is a gas, oxygen has to go higher, but it is not, it is glucose. So what happens is that glucose can go all the way up to here. The cells cannot go beyond glucose because if there is no glucose, they cannot grow beyond glucose. So they have to end at glucose level, but they can grow beyond oxygen level. So these guys, let's say oxygen goes up to here, glucose goes up to here. Okay, so this portion, because oxygen diffusion limit is here, this is low oxygen. Makes sense? It is low oxygen because oxygen is not diffusing there, glucose is going there, oxygen is not diffused there. Low oxygen is also called as hypoxia. Makes sense? So the tumor is hypoxic on the top where there is no blood supply. That makes sense? So think of it like in this situation. If there are two human beings, right? And we apply a lot of stress to both of the human beings, right? So this guy dies or collapses or gives up. But this guy survives that extreme stress, okay? So the guy who survives will become more aggressive. Now here we are talking about not good human beings, we are talking about tumor cells. So the tumor cell which will survive extreme environment like hypoxic condition, they are supposed to die, but they don't die. If they die, some will die for sure. Hypoxia, some will die, but some will survive. And if they survive, they are very bad. They are very bad. So angiogenesis, right? You asked about angiogenesis. So when you give angiogenesis inhibitors, tumor cells die because they don't get glucose, they don't get oxygen. But then some tumor cells will survive, and those guys will survive. They are much worse than the original guys. And I will tell you how they survive now. So they do symbiosis. Okay, so they make friendship with the guys which are getting oxygen, and they work symbiotically, and it is unimaginable what they do. Okay, so these guys are getting oxygen, so glucose is going all the way up to here. So this portion which is getting oxygen, they can have Krebs cycle, right? Because oxygen is there. So they can survive with low glucose because they eat small amount of glucose and they generate more energy because Krebs cycle is working. All of you guys are with me? So they have huge amount of, they have Krebs cycle. So because they make a lot of ATP, they use less glucose. If they use less glucose, obviously glucose will go higher up because they are not consuming these many glucose, right? So if suppose glucose is coming my way and I am at the top here, right? If you guys are not eating all the food, some will come to me, right? But if it is consumed in middle, it will not come to me. So these guys eat less glucose so that it goes higher up there. So in that way, they are helping these guys. What these guys are doing in return, now here they don't have oxygen or they have very low oxygen. So they can undergo glycolysis, but they cannot undergo Krebs cycle. So what is the end product of glycolysis? Pyruvate. And you know, when you exercise, what happens? You get these cramps, pain. What is that pain? Lactic acid. So do you guys know why lactic acid is formed? Why don't we stop at pyruvate state? No, that is anaerobic thing. But what is the reason lactic acid is formed? There is a very, see, I always tell my students, what's the reason? Why do we have to make lactic acid? The reason we have to make lactic acid is that what is the reaction, what is the overall reaction during glycolysis? We take glucose, if I have to write the net reaction, so glucose plus 2 NAD plus, correct? Plus 2 ADP, net reaction, not the input and output. Net reaction is 2 ADP. We get 2 pyruvate, 2 NADH, and 2 ATP. Remember net reaction. 4 ATP are formed, but we are also consuming 2 ATP. So once again, net reaction, glucose plus 2 NAD plus 2 ADP. Product is 2 ATP, 2 NADH, 2 pyruvate. What happens is that if glycolysis is going all the time, right? Like we are doing exercise, so that times for some time our muscles become hypoxic because we are vigorously exercising. So Krebs cycle doesn't happen. And you guys may know this thing or not know this thing, that we have a separate pool of NAD plus NADH in the cytoplasm, and we have a separate pool of NAD plus NADH in the mitochondria, and they don't intermingle. तुम अपनी चीज़ रखो, हम अपनी चीज़ रखते हैं. Clear boundaries there, okay? So what happens if glycolysis is occurring all the time? You have more and more NADH production. You don't have NAD plus, right? Because electron transport chain is not working, no oxygen. So eventually all NAD plus will get converted to NADH, and there's no way to regenerate it, correct? So glycolysis will stop, and if glycolysis stops, we die, especially under low conditions. So that is why the muscles have this thing that they convert pyruvate back to lactic acid. So pyruvate is reduced to lactic acid, so NADH is converted back to NAD plus, and glycolysis continues. And that is why we make lactic acid under anaerobic conditions. Is it making sense? To regenerate NAD plus, and I will give you an example to tell you how important it is to regenerate NAD plus. So suppose if we keep eating all the time, we are not even hungry, but every 1 hour we feel like munching something, so we are eating all the time. So what will happen under those conditions? NAD plus is always getting converted to NADH, correct? So we don't give it enough time to go back to NAD plus, and that causes toxicity in our body. So I will give you a real experimental example. They took 2 sets of mice, okay? They gave them exactly the same amount of food, but in one case they put the food in front of the mice. They could eat it all day long whenever they felt like. And in this case they gave it at certain interval, but same food, same amount, but you know, some hours apart. So these mice which were given food sometime apart, they had 30% longer lifespan than these guys who were eating all the time. Same food, total food, everything. Not healthy versus unhealthy food. Why? That is NADH toxicity. So you have to regenerate NAD+. That is why lactic acid is produced. So now I will tell you how tumor cells are doing the game. So they are surviving only on glycolysis. So they have to regenerate NAD+, so that glycolysis is going on. So what is the end product they are generating? Lactic acid, right? Now what they do, they give lactic acid to these guys. So what the lactic acid these guys are generating, they give it to these guys. What these guys do, they convert lactic acid back to pyruvate, and then they use it for Krebs cycle. Okay, so real symbiosis. They let the top guys have glucose, the top guys make lactic acid, give it to them. Now they don't need glucose because in one step they can make pyruvate, they can undergo. So they do symbiosis. So now they have evolved a real mechanism to survive. So they are even more aggressive. So these guys which are on the top, they are even harder to treat. So that is why angiogenesis inhibitors is a double-edged sword. Answers your question? Next question, yes. Will come this way. Okay.
Can you go in brief how mental stress is related to cancer?
I can go in huge details. Sorry. Just stress is one thing, okay? Stress hormones actually destroys everything in our body, okay? So stress is manifested in many, many ways. So when we are facing stress for whatever reason, the stress hormones can change our metabolism, okay? Some people eat too much during, some people don't eat too much. Usually people who have stress, they don't exercise, they have obesity, they have other issues, right? So all of these things will contribute to diseases. And also stress can cause a lot of acidity in the body, and acidity favors cancer. So you can actually give real metabolic reasons for that. I mean, stress is a root cause for diseases. Real root cause for diseases. So the way I understand human body is that, our human body is made in such a way that you cannot consume too much food. If you consume too much food, it will become unhealthy. You cannot really fast too much, it will become unhealthy. You have to exercise. You cannot eat the food you really love, right? The kind of food we love is not good for our body. That is the truth, right? We like ice cream, no, bad. If we want to have samosas every day, bad, right? So it is just like everything is in moderation, the way it works the best. I don't know why it is that way, but it is the way it is. And there are so many things which will directly lead in the science field, stress to cancer, to obesity, to neurodegenerative disease. The brain actually degenerates. The brain actually degenerates due to stress. The cells in the brain, they die. Stress can lead to diabetes. So, and in diabetes, one of the treatment for diabetes is exercise. When a person is diabetic, you say the person needs to exercise. That's the first thing they say. I can tell you what happens when the person exercises. Serotonin and everything, that's one thing. But when you are exercising, your ATP is getting converted to ADP. ADP is getting converted to AMP. Makes sense? Now, our ATP levels in the body is about 5 millimolar. ADP is some very, very low amount, like 10 times less than ADP, even less than that. AMP is extremely low in our body, like micromolar. So anytime AMP levels increase just a tiny bit up, all the cell signaling is going, oh, AMP is going up. So there are many enzymes which get activated when AMP levels go up. So one of the most famous is AMPK kinase, AMP dependent kinase. So that kinase binds AMP, and kinases phosphorylate different substrates, right? So AMPK activation, and when it is activated, it upregulates those pathways which helps in diabetes. Okay, so metformin is actually an activator of AMPK. So without exercise, it is activating that guy there, AMPK. So there is actually, the whole pathway is mapped out to know. It is exactly known. This was discovered by my friend at MD Anderson, that how metformin is an anti-cancer agent. He did not discover this, but he showed that what dose of metformin is anti-cancer. Okay, so we have the pathway mapped out for that. So for stress, we actually have the whole pathways, the players and everything, how it leads to cancer and diabetes and neurodegeneration. But go ahead.
I wanted to ask the same question, like role of modeling or using advanced approach in designing.
Yeah, so this is a very good question. So can you repeat it so that because I think the question she's asking is good for all of you. It's a little bit away from cancer, but very relevant question. So can you repeat the whole question a little bit loudly?
Yes, ma'am. So my question is, ma'am, there are many drugs which are already approved, which are designed based on computer-based drug designing approaches. For example, we can see zolmitriptan for migraine, which is approved in 2003. Then dorzolamide for glaucoma and ocular hypertension, which is approved in 2012. So I wanted to know your opinion, like what is the effect of computer-based drug designing and actually going for designing any drug, like for example going for designing then synthesizing, or going reverse, that you already get the experimental data and then support it with this prediction based.
Yeah, so the basis that she asked me this question, may I have your name please?
Janvi Rao.
Janvi, right? Yeah, so the basis for this question is that I have worked at Novartis, which is a pharmaceutical company, right? So she was asked, so this question is very relevant. Do you do modeling first to make a compound and then you check it experimentally, or you do experimentally and then do modeling, or which stage you should do modeling? So I will tell you my own experience here, okay? So a long time ago when I was a postdoc, right? So we worked with kinases, and I was a chemist, as Venkatesh said. So I was an organic chemist who had zero knowledge of biology. So at that time, there was another student in our lab who was a graduate student. So he did some modeling, and so we were trying to make, kinases are a set of enzymes that use ATP. We'll spend some time with kinases also in this course. So those are those enzymes which bind ATP and phosphorylate different proteins. To make a long story short, modeling suggested that we should make this kind of ATP and it will work extremely well, right? So I was the chemist, so he just suggested and I made it. So I made about 50 different analogs. You guys know the structure of ATP, right? So I will tell you what exactly he suggested and what I was making. All of you guys can see this side of the thing? Okay. So you know ATP has this adenine ring, right? And there are 3 phosphates here, right? You guys cannot see the 3 phosphates, but there are 3 phosphates. So he predicted that if I put some huge group here, this was the exact compound. If I make CH2-phenyl, that would work very well in our system, okay? So it was very hard to make, it was not a benzyl group, it was sulfur here and then CH2-phenyl. So he was very good in modeling, and he suggested if I make this compound, it will work very well. So that was very hard to make because I had to do a bomb reaction. So because I had to heat it so much that I had to use a bomb kind of thing. That was the only thing. It had this thickness. and I would put the reaction and seal it and then heat it for 6 days. And I was a very good organic chemist because IIT Kanpur really trained us very well, but my yield was like 10%. So it was a very bad reaction, not my fault, bad reaction. Okay, so I hated that reaction because you do it for 6 days and your yield is so poor. And he said, this is the compound which will work. So I made it and it failed completely. So then my advisor said, why did it fail? So what happens is that this guy's prediction was not wrong. Okay, proteins are very smart. So when I made this compound, so the idea was that protein pocket is like this. So an ATP binds this pocket. So if you put such a bulky benzyl group here, then it will not fit in the pocket. So we had to make a ATP analog which would not fit in the pocket. So this guy did exactly the right prediction. But why did it not work? It was accepted by the kinase, by the enzyme, which was very shocking. You're putting such a huge group. How can it be accepted? So it happened that there was a phenylalanine here, and phenylalanine has CH2 phenyl group, right? That's exactly what I was pulling. So they should crash, steric thing. It should not get accepted. Proteins are very flexible. So if you put that phenylalanine would just reverse 180 degrees, and now that side group is facing outside. And it accepted it and it failed. So it made me very happy that I don't have to synthesize that compound again. But the prediction was wrong because the protein switched. So then I made all the compounds which were made at NH2 group here. That was a piece of cake to synthesize. And I made 50 compounds and they are still used worldwide. It is so powerful method. Okay. So to answer your question, it didn't work because we started with modeling first. Now I have met a lot of people who do modelings, right? But they have no way of testing. Maybe some compounds will work, some compounds may not work. But now I will tell you another side of the story. So, and this always works, what I am going to tell you. So, in my experience, what industry does, so industries have millions and millions and millions of compound libraries, right, like Novartis. So, they will screen millions of compounds, actual screen, and let's say they get 200 hits, okay? Then they will rescreen it, so you get more, like you reduce it to some 150 or 100 compounds. But getting a hit is not good enough. You have to do selectivity screening, that it is selective for your target. Now you have reduced it to a lot less. Now you actually do the SAR because you have methyl group here, ethyl group here. Methyl works a lot better. If you make isopropyl, it doesn't work. But when you make propyl, it works. Wow. So it seems it's linear. You cannot put a branched group there. So you do that kind of SAR. At that stage, once you have some experimental data with the SAR, that's when you really need modeling. You really need modeling. And then you see with the experimental data, that's where modeling is extremely powerful, okay? And then modeling can guide you because, and then you can actually check your modeling with the actual data, and that can do wonders, and you can make a very powerful compound. And now I'll tell you the actual success story. I was telling this story to someone a few days ago. I cannot remember that lady's name. She's an Indian lady, she was not famous at all at that time, okay? So she was working in this company called Plexicon, okay? And she had just one technician working for her in an industry. So Plexicon is a pretty decent, very good company, but she had just one person working with her, that too a technician. So they were doing things together. So she asked him to synthesize some compounds or high throughput screening, and they got some hit. And it was like, in a huge company, you are running your one-person lab, nobody cares that much. But she was working against a very important kinase, which is a very important drug target. We'll come to that target also. This is RAF kinase, RAF. So in cancer, there is a mutation in RAF, which is V valine is converted to E at 600 position. So it's V600E RAF. So she was working against that. So she got a hit, this thing. So she did some SAR and then she did some modeling. So this lady knew how to do modeling. And with her technician, they did some modeling. Based on the modeling, they said, oh, we should modify this compound this way. This technician was an organic chemist, so he modified it. And that became a $1 billion drug. One person team made that company billions and billions of dollars. Okay, she's a very famous person. Okay, so modeling can do wonders. So the take-home message is that they have to go hand in hand. You can start early, but I would start with the experimental data first. Because proteins are smart, they can change themselves.
She is interested more in computer doing things.
Yeah,
which I was
don't
telling
start,
her
yeah.
that you make compounds, do the screening,
And then do the modeling, and then it will do wonders for you. Yeah, last time when I came, not last time, in 2016 when I came, we were in Silver Spring at that time. IIT campus was not built. So one night, somebody came from Indore, not from IIT Indore, but some school from Indore. And they had done a lot of screening, from virtual screening. And they had a lot of hits. And they wanted to know, like, whether it is experimentally possible. So, but it is very hard to know whether it is possible. You actually have, so they were asking my help, but I don't work with those proteins and things like that, so it didn't pan out. But what I'm saying is that virtual screening has no significance unless you do the experimental thing, because you actually have to go to the biological systems. And then when they talk to each other, it becomes very powerful. So, this is a very nice thing for all of you, right? You need to use both synergistically. Okay, next question, please. Yes.
Ma'am, so a study said that in India, every 1 out of 5 women go through PCOS, like in the recent times. But it's also said that PCOS is being linked to endometrial like cancer. So how do we
PCOS?
polycystic ovarian syndrome?
Okay, polycystic, and that is linked to what?
Or endometrial cancer, ma'am.
I don't know any details about that.
There have been like multiple studies which say that it is being linked to that. So I just wanted to know, like, how is it being linked to, like, how can a hormonal imbalance lead to such a, cancer? Yeah.
I can give you many reasons how can hormonal imbalance. I don't know about this particular case, but I can tell you tomorrow for sure exactly, because I have to see what is what disease, because I don't even know much about the PCO disease. But hormonal imbalance can easily lead to cancer, okay? Because what happens is that when I'm just talking in a generic term, when hormones, they are very useful for our metabolic pathways, right? Insulin, we all know how important is insulin. So hormones are released by, what are hormones? How would you describe them? They are released by pancreas, somewhere here, controlling every cell of our body, right? Released here, controlling every cell of our body. Released pituitary gland, controlling every cell in our body. So they work, and they work in, throughout the body, depending on what kind of receptors they are binding to. So hormones in general control all the metabolic pathways, and cancer is hugely controlled by metabolism. We don't have time to go in cancer and metabolism, but the thing is that, like I said, before 2004, there was nothing like cancer and metabolism. Cancer was not considered to be a metabolic disease. Diabetes is metabolic disease. We knew it for a long time. But cancer was not considered as a metabolic disease. Now, cancer and metabolism is the hottest field, is one of the hottest field, right? So, hormonal disbalance can lead to metabolic problems that can lead to cancer. Easy connection. I don't know what is the exact situation because I don't even know much about PCO. So, but I can tell you tomorrow all the details. Once I know what exactly is PCO. Oh, it is PCO.
PCOS or PCOD.
Okay, so I didn't even know that. Yeah, tomorrow I can tell you PCOS or PCOD. Okay, yeah. Yeah.
Ma'am, I asked you one question at lunchtime, like related to target therapy.
Yeah.
Ma'am, abnormal receptor are increases in cancerous cell.
Yeah.
That time, receptor are blocked or mutated.
Yes.
Then that time, if ligand are not enter in cell, that also were related to Venkatesh in CRPC, prostate cancer. Then how to cell change the signaling pathway when ligand not reach the nucleus receptor? If already they are receptor are blocked or mutated by doctor scientist, then how to know cell they change the pathway?
So your question is that how do cells know how to change the pathway,
Yes.
or how do cells change the pathway? These are two different questions. So,
Ma'am, like if androgen is blocked,
yeah,
cell is not proliferated, but after some time at the
they become resistant, and how do they?
resistance.
Yeah.
Yes, ma'am.
So that I can tell you, right? So I gave you this example of symbiosis, right? So normal conditions they use oxygen, but when there is hypoxia condition, they change the signaling pathway. They just use glucose, they give lactic acid to other cells, and they keep using. So they downregulate Krebs cycle, right? So this is just one adaptation method. So now I'm going to come to the specific case of prostate cancer. So those of you who do not know prostate cancer, the main driver is androgen. Okay, so prostate cancer cells have androgen receptors. They eat androgen. Signaling pathways are upregulated, and in prostate cancer, it's hugely upregulated. So androgen receptor is overexpressed, and that leads to prostate cancer. That is the main driver. Many other things go wrong, but that is the main driver. So you get a lot of androgens. So when you give therapy against androgen receptors, first thing they do, mutation. So now they don't even need outside androgen. They can make their own androgen, or they will mutate it such a way that without androgen also the signaling is active. So you still block them by different ways. So now they know that they cannot depend on androgen. So now they will downregulate androgen signaling, and they will express new proteins. One protein is Aurora A, like Professor Venkatesh was saying. So when Aurora A kinase is overexpressed, you don't need androgen receptor. Aurora A alone is enough to cause cancer. So, go ahead.
But that time, ma'am, this shift from local hormone. If you block the neuronal hormone like androgen,
No, I'm not
estrogen.
even talking about hormones. So you are not even giving, forget about neuronal hormone or everything, right? So now they don't even need androgen receptor, they don't need androgen. They make a new protein called Aurora A kinase. And Aurora A has nothing to do with androgen receptor or androgen. Aurora A alone will cause cancer. Aurora is expressed in huge number of human cancers. So Aurora A alone, so now these cells don't even care, like a completely new kind of cancer, still prostate based, but it depends on a completely different driver, and Aurora A kinase. Then when I was telling you the neuronal phenotype, they express some proteins like synaptophysin, they change their whole thing. So what happens is that, now you will say how they are expressing Aurora A kinase, what is leading to how they are doing it. So what happens is that it can happen at many levels. Either, I will also talk about those kind of things, why some protein is overexpressed. What happens is that when a protein is made, it is always a balance of birth and death. Okay, so death is also equally important as is birth. Okay, because everything has to be balanced. So when a protein is made, no matter which protein is made in the cell, right? So some proteins are like in this amount, some proteins are in very low amount under normal conditions. So the guys which are in low amount should remain in low amount for under healthy condition, and the guys which are in huge amount should remain in that level. But what exactly is this level? This is the steady state level of a protein, and that is a balance of protein synthesis and protein degradation. Makes sense? That is why it maintains this level, whether it is low or high. But suppose it is not getting degraded, then the levels will rise because protein synthesis rate is still the same, but it is not getting degraded. So RORI many times is not degraded in cancer cells. So the levels steadily increase. So DNA is amount the same, RNA is the same, but the proteins are much higher because it's not getting degraded. We have published many pathways which shows why RORI is not degraded. And there are many ways in literature which shows why Aurora is overexpressed. Or sometimes it is possible DNA is amplified and you make a lot more RNA and you have a lot more Aurora protein. So there are many ways that the protein is overexpressed. So when you block androgen signaling, it is known that other oncogenes are those proteins which can cause cancer. Other oncogenes are overexpressed. Another protein which is overexpressed is MYC. So MYC is a very unstable protein, but when Aurora is there, MYC becomes very stable. And these two can take the charge for cancer. They don't need anything else. So cancer cells, they adapt very quickly. Now I will tell you third reason. So your DNA is the same, right? But DNA needs to be told whether it should make the RNA or not. Okay? So this comes under epigenetics. So not all the DNA makes RNA. So there is a process called DNA methylation. So if you methylate the DNA becomes very tight. Okay? And that does not make the RNA. RNA polymerase cannot access it. When DNA is not methylated, it is very open, it makes the RNA, right? So, or histones modification. These are all epigenetics. We'll discuss that later on. So, under these conditions, so what is epigenetics? It tells you whether it should make the RNA, and if RNA is made, protein will be made. So, DNA is exactly the same, but normal cells have different message about this DNA. Cancer cells will have a different message. So, this will be expressed in cancer cells, same DNA, but this is not going to make protein in normal cells. So there are many epigenetic things which are changed in cancer. So that is why the players are changed. So imagine that a company is run by a group of people and those people are targeted. New group of people come. They can even take it higher. But these are new guys. They will change the policy, everything. They knew what was not working last time, so they're going to change, right? Suppose, in my time when I was new in this, in US, travel agencies were very popular, right? Every a second shop was a travel agency because we had to depend on them to book the tickets. And then, even 20 years ago, travel agencies start, disappearing. Now there is probably 1 or 2 travel agencies. I mean, we had a bad experience. That's why we are laughing, because we had to go through travel agency in this time also for my tickets.
Yeah, in India only 2.
Only 2. And we used to have, every second shop was a travel agency in one time. It was so popular, right? So you have to adapt according to the market, right? So androgen not working, bring new sets of guys, right? Cancer cells are very smart, very adaptable. That's why this disease is just, it keeps on evolving. But, it is, trust me, we have a better hand than what we had 20, 30 years ago. And day by day, we are going to become better and better. We have eradicated many diseases, which were very big things in the past. We are going to eradicate this disease also someday. So, yes.
We have different type of kinases, limb kinase, aurora kinase, CDK kinase. So is the activity of one kinase is related with the another activity
Absolutely. So it is, so these are the 3 kinases we work on in our lab. CDKs and limb kinase and aurora kinase. So aurora kinase activates limb kinase, LIMK2 activates aurora kinase. One kinase can inhibit other, the other can inhibit the other, or one can activate the other can inhibit this. So kinases are actually, there is a huge network, they're always talking to each other, huge crosstalk, direct and indirect.
If we target one kinase, then it will affect the other kinase.
It is possible. I'm also going to give you polypharmacology. There's one lecture on polypharmacology. So what people used to think, remember I said specificity, when you use small molecules, it's not that specific. But sometimes having a dirty drug can be very good, because if your dirty drug targets many bad culprits, like many culprits at once in cancer, that is much better than one drug targeting one culprit. So many dirty drugs are very good because they are hitting all the right targets. Okay, so first people used to say magic bullet, right? Now they say magic shotgun, that it is the magic shotgun which is spraying the things all on the bad guys. And real life example is the most famous drug in kinase field, Gleevec, right? So Gleevec was the first kinase inhibitor which was FDA approved, and this is for CML, chronic myeloid leukemia, and it was against BCR-ABL. But if you inhibit only BCR-ABL, it is not that good. And this Gleevec hits PDGFR, c-KIT, and because it inhibits these 3-4 targets together, that is why it is such a wonderful drug for CML. So sometimes hitting more than one is much better, and that comes under polypharmacology. So we discussed that too in this course. Yes.
In targeted therapies, cell receptors, it's like mutated cells have multiple receptors, and we use drugs to block those growth receptors. So now, how does our drug differentiate between mutated cell and normal cells receptors?
Yeah, so you can imagine that normal cells have this shape receptors and mutated receptors are like this. So when you make the drug, you make it for this one, not for this guy. But it is also possible that both cancer cells and normal cells have the same receptor, but cancer cells have a lot more receptor. So you will inhibit cancer cells because they have more chance of getting inhibited. But you are also going to inhibit normal cells, right? So two consequences. In one case, normal cells don't need that receptor. So even if you inhibit it, not side effect. The other consequence is that so in that case, cancer cells really depend on this receptor, normal cells don't, which is very likely, right? So not too much side effect. But then there's a possibility both like their own receptors. So when you're killing cancer cells, you're also killing normal cells, side effect.
The mutated cell has, cancer cell has many receptors. So if one receptor is blocked, but the cancer cell generates self-growth hormone signals, so it can use its other receptor to generate the signal.
So, but if you are yeah, I understand your question. So your question, you are asking 2-3 different possible scenarios as I understand it. So cancer cells have many receptors, but all these receptors are of the same kind. Okay, so when you inhibit, and if they are generating their own growth receptor, then they can inhibit. But keep in mind, if your inhibitor is more potent than the ligand which binds here, the compound will outcompete the ligand. So even if the growth hormones are there, your inhibitor is binding much more tightly. So that will stay there despite the growth factor being there. So you have to make a much better inhibitor in that case. And sometimes what you do, instead of binding the receptor, you can even stop the growth of the growth hormone. You can inhibit the enzyme which is making the growth hormone. So now there is no growth hormone. This receptor will starve. Right? So there are many ways to do that. Yes. Your turn. Yes.
In regard to what she asked about modeling compounds.
About what?
About modeling of compounds.
Compounds,
So
yes.
So I do computational work, and the question that usually comes up is that what is the rationale of you making your compounds or coming up with the compounds?
What is the
Rationale of making the compounds or modeling the compounds. So normally we model it according to the ligand. If the ligand is known, it binds to the particular protein, or if you know the structure of that particular target. So you model your compounds according to either the ligand or the protein. So that you now say that you made your compound in regard to
You
the
can do
ligand
both actually.
and the
You can do
structure.
both. So suppose you know the ligand. So what is the difference between a ligand and an inhibitor, right? So let me say, so I will give you an example. So let's say the kinases, right? So kinases, what is the ligand here? ATP. It binds ATP. So if you make something like ATP, right? If you just modify the ATP to make it bind more tightly, but it is still ATP, so it's still going to transfer the phosphate. So you are making yet another ligand, you are not making an inhibitor. But if you make some compound which will fit in the pocket more tightly than ATP, so it has no phosphate, so it's not going to do the job, but it is blocking the pocket. And when it is blocking the pocket, ATP will no longer bind there. Makes sense? So if you make an inhibitor, now sometimes kinase inhibitors, often have purine-like structure. It has adenine rings, so it may have purine-like structure, but not exactly purine. The nitrogens are not there, but there's a bicyclic flat ring there. And then you put different kinds of groups all around it, right? So it has some resemblance, a little bit resemblance to ATP, but very far away from ATP. It does not have the sugar thing. It does not have the phosphate. So to answer your question, usually people start with some kind of pharmacophore, and then they modified around it. And then they first do SAR, whether it is even worth pursuing it. And once they get some data out of that, then they do modeling and modify it further. So usually, suppose you are in an academic lab. So what you do, based on literature, you make some compounds, not too many, maybe 20 compounds or even 10 compounds. And then you screen them to get some idea, right? And if you have even some positive data, you can build around it. But here the activity, the structure-activity relationship is extremely important for your guidance, because you will often see 1-methyl to ethyl to propyl to isopropyl to butyl to tertiary butyl to isobutyl. You will get huge amount of information. Put a fluoro group here and there, right? Hydrogen bonding, it's just huge information. So that can guide your thing. But if you want to make a I know of many people who have just randomly made library of compounds, and they have tested it, and sometimes it works like magic. I know of a person who made a library, a purine-based library, and it got published in Science because the compounds were big hits, and it's a landmark paper. So basically what it was, they didn't have a fixed starting structure. They looked at the protein pocket, binds ATP, let's make purine-based library. And it was just a huge hit. And then the whole world used these compounds as models to further modify them. And the other way to go around is that you can do some screening. Again, not just virtual screening. I would not do virtual screening. I would do actual screening, which is very easy to do, and get some guidance there, and then do the modeling thing.
Okay, another question regarding you mentioned about the breast microbiome,
Yeah.
and how it interacts with the environment and genetics. So I know of the gut microbiome, but the breast microbiome, what composed?
About which
The
microbiome?
breast microbiome, breast.
I don't know exactly what is breast microbiome there, but I can guess that when breast microbiome is dysregulated, it can give rise to, microbiome can really contribute to cancer. So if it is localized at some part of the body, it can really create problem in that part of the body. So it can contribute to some extent to breast cancer, but usually that alone is not sufficient. It cannot trigger it from the start and lead it to breast cancer. But if there is some propensity, it will help it to develop breast cancer. So suppose it kills the immune cells or it inhibits the immune signaling in the breast, so that person is more susceptible to have cancer there. So they increase the susceptibility of cancer, either prevent it or promote it depending on what kind of microbiome is there.
Because the microbiome is the microorganisms, like in the case of gut, there's bacteria.
Say that again?
Like in the case of gut, there's bacteria, which is the microbiome. But so for the case of the breast, which kind of microbiome is there?
So usually overall 97%, more than 97 is different kinds of bacteria. But it could also be viral infection which can directly lead to cancer, and that is also microbiome. So there are many viral based cancers. Your question, yeah.
The main limitation of tumor or cancer therapy is relapse. So which is mainly because of like cancer stem cell. So like because they are unresponsive. So like we have discussed several clinical strategies. So is there any like targeted strategies to directly target cancer stem cell, because these are the main reasons for relapse case?
This is absolutely true. So do all of you know what are cancer stem cells? You don't know that, right? So I will tell you, this is also a topic we will cover eventually in this course later on. So what happens is that suppose somebody has this big of a tumor, right? So this is the whole mass of the tumor, and you give radiation therapy. So when you give radiation therapy or chemotherapy, it is possible that you will kill 99.99% of the tumor completely. But you will have actually 1 or 2 cells remaining. Imagine from trillions of cells, everything has died but 1 or 2 or 3 cells. Those are the resistant cells. And the guys which are resistant, they are the real aggressive one, as I keep saying. If they survive therapy, they are very bad cells. So what are cancer stem cells? Do you know what are stem cells? You guys know what are stem cells? Yeah, don't worry about that, okay? So stem cells, so you know how human or any embryo during embryogenesis, right? What happens, we start with zygote, egg and sperm and zygote, one cell, and that one cell divides and makes the whole body, no matter what kind of body it is. So those cells have the capability to make any organ in the body. They are rich in stem cells. So when I joined Novartis, my director Peter Schultz said, "Oh, we have to find a way to make a constant supply of stem cells." So if we have that time nobody had any idea. You guys have 2 full lectures devoted on stem cells, and that is a very huge part of this course. I've spent like huge amount of time on those 2 lectures to make the stem cell story very solid. So what happens is that imagine somebody had some kind of cancer and they cut out a whole huge part of that organ, right, to make that person survive. So if you have stem cells, you can regenerate the whole organ, right? When you do, many times people remove bone or part of the whole body during cancer therapy. If you have stem cells, you can regenerate it. So now there are ways of even taking simple like skin cells, right? These are not stem cells, just the regular cells. And you can make them stem cells. We know how to make them stem cells. And if you have stem cells, you can make any cells. Out of the skin cells, you can make neurons, you can make liver cells, you can make anything, right? But in our body, there are some stem cells. Even in adult body, there are some places their stem cells are there. So you can take those stem cells out and you can regenerate them to use to make some organs. However, they do not have infinite capability. So for example, if the stem cells are somewhere from the blood, so those stem cells can only make blood kind of cells. They are not going to make the bone. Okay, so the ones which are close to the bone, they are more prone to make similar kind of cells, not one but similar kind of cells. So they can make limited number of organ, but not all of them. But when it is the embryo, it can make anything. So what people thought, let's do in vitro fertilization. We make egg and sperm, and when the embryo is in the blastocyst stage, those embryonic cells have infinite capability to make any kind of cell in the body. But then there is a lot of ethical issues. If you do in vitro fertilization and you kill the embryo at the blastocyst stage, is this ethical? So that is not allowed, because it's always life and death situation, whether this is alive, whether it is not alive. So to avoid that, now we use iPSC cells, which can make limited number of it, but you can still make. So you can either take non-stem cells and make them stem cells, or you can take these stem cells which exist in the body with limited capability and you can do it, right? By the way, I've forgotten the question in all this stem cell thing. Yeah, cancer stem cell, right? That was the question there. So the stem cells can make, so if coming back to cancer stem cells, these 1 or 2 cells which survive, 1 or 2 cells, they can go and they can go to some other part of the body. So let's say they're epithelial cells, and epithelial cells are like brick, so they cannot move through tiny holes and go out. So these epithelial cells, cancer stem cells, they become mesenchymal cells. So epithelial cells are like bricks, mesenchymal cells are like this. So they will change, and this process is called EMT, epithelial to mesenchymal transition. They will become very thin and they will go through these holes, metastasize to some other part of the body, and there that one single cell can form the whole tumor. And their resistance to chemotherapy, radiation and everything. So now we know the signaling pathways by which they are so resistant. So people are actually making therapy against cancer stem cells. We know which players are the one which make them undergo this epithelial to mesenchymal transition. So if you target those proteins, these cells are susceptible to death. So cancer stem cells are extremely, extremely, extremely resistant. But people have figured out a way to even kill them eventually. But the bottom line from all of this discussion is that cancer stems can adapt very easily. They can become resistant very easily. So you have to be very smart in killing them as quickly as possible. So even when the therapy is working, people are not very hopeful, because you never know when they're going to adapt and become resistant. And that's the biggest fear all the time. Go ahead.
Ma'am, in targeted therapy, you said that some patients' own immune cells were withdrawn and then engineered and re-injected. So my question is, does that mean that the immune cells behaves differently from patients to patients?
No, so the thing is that there is a lot of things there. So what happens is that if you take it out of the patient, so they are called allogenic versus what is the other word? Autologous versus allogenic. Yeah, thank you for reminding me. So what happens is that if you take it from the person, same patient, and then you modify them, it is like you take them and then you give the right weapons to those T cells and you put it there. Because it is derived from the same patient, the immune system will not get activated, because it will recognize it as self, because it is from the same thing. But if it is from a different person, there may be a severe immune reaction, and that can even kill the person. You know that when there is organ transplant, there is a lot of matching. So that is why it is taken from the same patients. But then we know what are the things which are different between patient to patient to patient. So you can take T cells, and those things which are different, if you knock that gene out, now immune system cannot recognize it, because they have the recognition based on those proteins, and those proteins are not even there. So now it is like a shelf thing. You can just make CAR T out of that, and you can give it to any patient. Make sense?
In colon cancer, there has been seen that it developed from mostly from IBD. Means,
Please repeat the question.
in colon cancer, when developed, so it is seen that according to me, what I read, that it mostly developed from IBD, means ulcerative colitis, and
Yes, that's correct.
so there is also means colon cancer happens by direct mechanism, or it totally depend on IBD.
I don't know that much details about. Tell me the full form of IBD.
Inflammatory bowel disease. There are two types: Crohn's disease and ulcerative colitis.
Yeah, so there is a huge link between inflammation and cancer.
Yeah.
Colon cancer is one of them, but inflammation is invariably linked to almost any cancer. So in any part of the organ, if there is a chronic inflammation, the chances are it will lead to cancer. And I can explain to you in very simple words, right? So what happens when there is inflammation? When there is inflammation, suppose somebody gets injured, right? And whenever you are injured, there is inflammation in that place. Why there is inflammation? Because all the repair cells, they come and they try to, cover the thing up. It is a very quick process. So whenever they cover the injury, it's never smooth. There is always some kind of roughness there, right? And it takes a long time to become smooth. Why it is like that? Because when there is injury, they come, they patch it up, and they're really in a hurry to patch it up. So it's not a very clean job. Okay, now imagine inflammation is constantly persisting. So they are constantly working on top of each other, and I'll show you slides of that also. So what happens? Constant bringing in new cells, not arranged properly, leads to cancer. So colon and all of these things, digestive system, they are very prone to this kind of thing, because they are actually involved in mechanical stress, right? And mechanical stress, and then inflammation, and then repair. So they become more prone to cancer under these conditions.
And there is also seen that in Western countries, there are occurrence of these IBD cases, ulcerative colitis and Crohn's disease is more as compared to India and Asian countries. So I have read one is the lifestyle changes that happens that cause, but what is the other?
I don't know about that. I mean, I can speculate. I really don't know. The thing is that I was very naive in thinking that all cancers are the same, and if you work on one cancer, it is the same as other. It is not. Every cancer has its own story, and they're as different from that. So I don't know much about colon cancer, but lifestyle changes. So the thing is that lifestyle is very different from person to person to person, even in the same country. Okay. So, obesity is more common in Western countries than in Asian countries. Now, people are obese here also relatively, but it wasn't the case before, because, there were no computers, so there was no desk job, right? If there was no desk job, even people who had very good jobs, they were also moving all the time. Right? People were more social because there was no television. So people were not sitting in front of television and eating popcorn, right? They used to go and meet friends or meet outside. So lifestyle was very different, and it was changing. So lifestyle here is different from the lifestyle in some other place than other place. So, and then people are different. Some people are active, some people are not active. Food habits are very different. Okay, like I was saying earlier, I don't have the actual data, but I know India has a lot less Alzheimer's patients than in Western countries. So part of it is that people live longer there, and they live past 90 pretty easily there, and they're more prone to Alzheimer's because of the age. But I also personally think that eating the kind of food that we eat here prevents Alzheimer's disease. So it can be applied to colon cancer also. And then I don't know, this is again speculation, that maybe eating a lot of non-vegetarian food can lead to cancer. It is also possible, again speculation, that if you eat meat, right, and if the meat is infected by viruses or something, that can lead to disease. So it depends on many factors. It could be also that they have genetic predisposition towards this disease. So this is a known factor that, some population, like people from China and similar countries, they lack alcohol dehydrogenase enzyme. So if they drink alcohol, their face turns red. Have you seen or have you met such person? You guys have not met. My close friend has this thing. So whenever he drinks even a little bit of beer, his face turns completely red and he starts sweating. So when we were students, every Friday there used to be TGIF. So thank goodness it's Friday party. So all the students, used to be a lot of drinks, Cokes, and that time we didn't know Coke was so bad for our health. But as students, you can eat a lot of stuff because you're working so hard. So everything is, you don't get the side effects. So Coke and beer. So he used to drink beer and his face used to become so red, like half a bottle of beer and he was just totally red. But we don't have that deficiency in India. And it is not there in European decent people also, but it is. So I don't know if they're prone to those diseases because of some genetic predisposition also. Make sense? Yeah.
My question is not close to what we learned today. It's not related to what we, but in some way it is very close. I was wondering why is it that animals. I was wondering why
I have to come closer when you speak.
I was wondering why is it that animals do not get cancer, yet they have the potential of getting cancer. For example, we are using mice. We can induce cancer in mice and use our drugs to see whether it has effect on cancer. Yet on their own, they are not able to get cancer. And we know that in biology we have conserved genes. It's conserved gene in animals and humans.
So,
and they have the potential of developing. And we witnessed that one in mice.
So I can answer this in many ways, right? So if I understand your thing, that we induce cancer in mice, but mice don't get cancer on their own, right? So one of the reason could be, what is the life span of a mouse? Do you know that? I don't. How many years?
3 to 4 months mice.
They die in 3 to 4 months? I don't think so. No, there is no way, because we have Alzheimer's mice brain in our lab, and if you induce Alzheimer's disease in a mice, you can collect it even when the brain is degenerated. You can collect it after 15 months. So normal mice definitely live past 2 years. But let's say they have 3 years. I don't know what is the lifespan of a mouse. Of a healthy mouse, maybe let's say it is 3 years or 2 years, whatever. What is our lifespan? So we collect a lot of damage over our lifetime, and that makes it a lot more susceptible. The mice are not smoking. Humans smoke, right? Mice are always running around. They're super active. We don't exercise, right? So mice are much better than us in many ways, right? That probably keeps them protected, right? Mice is always running, very healthy. I can also give you a specific example, right? Mice never develop Alzheimer's disease. So other than the age factors, mice actually lack the genes which predispose humans to Alzheimer's disease. They don't have those problems. They don't have those genes. So Take home message, exercise. Okay? Answers your question?
So if they don't have the genes, why is it that they develop when you stimulate?
Say that again.
When you stimulate the cancer, it develops. If they don't have the genes, why does it
No,
develop?
so when we do Alzheimer's things, we actually put those genes there. So we don't add the genes in the mice, but we inject them with cells which have those genes. And then we induce Alzheimer's-like state in them. Dogs, they develop cancer all the time. You know that. They spontaneously develop cancer. But mice, they don't. Part of the reason lifestyle, so, yeah. Yes?
Can cancer affects the progenitor cells, like in hematopoietic stem cells?
Oh, absolutely.
They do?
Yeah.
And they can create an army of the defected cells, and they might...
That is what is leukemia, right? Bone marrow cells become cancerous cells. Yeah. You know, I don't know who I was talking to. I get confused because I talk to many people at the airport also. Yeah, I think this was an airport guy. So his father-in-law has leukemia, and he lives in Indore. So we were both coming to Indore, and he was diagnosed with leukemia right after COVID or during COVID time. And the doctor said he has leukemia killed Rishi Kapoor, right? He went to New York for 1 year, got so many transplants, still came back to India, and after a few months he was gone. So leukemia is pretty bad disease, but his father-in-law has a kind of rare leukemia where he's completely fine, free of cancer, and I hope he stays that way. So they are celebrating their 50th marriage anniversary this month. So, yeah, there are many different kinds of cancer also, rare cancer, aggressive cancer, less aggressive cancer. And I was never working on leukemia, so I was not that interested in leukemia, but I have met a person who was very close to me who had leukemia. So I had this undergraduate student in my lab. She was like the truly outstanding person. It is hard to believe how outstanding she is. So when she applied to my lab, she said, you know, you get like 10 applications a week, so you don't pay attention to those many applications. But she wrote, she has already worked in Google as the head of a whole team. She has flown, she was from Singapore, she has flown Singapore Airlines plane. She sent me the picture in the airline thing, sitting in the cockpit. Okay, she said she has worked in the World Bank, and she has led a team in the World Bank. Google, World Bank, plane. And she was 21 years old. Is it even possible? And that is the truth. She was a miracle child. She was diagnosed with leukemia when she was 2 years old. So she had 2 or 3 sisters who are all younger to her. So her parents said all the sisters always have to be together so that they can take care of the elder sister who has leukemia. So when I met her, she was obviously a very young girl, but she used to wear a lot of makeup on her face. So like huge amount of makeup. So I used to think why she's wearing so much makeup, but obviously you don't say this to any person, right? Whatever it is. I didn't know any of her history, medical history. So, and then one day, and so so the undergraduates, they go home in summer, right? They don't stay in summer. So one time she stayed during the summer in my lab, and halfway through the summer she said, I have to go to Singapore, I have to take a therapy. So I said, what therapy are you talking about? And that's when I came to know she had leukemia, and every 3 months where I live, she used to go to the hospital, get admitted, and have I don't know what therapy. Again, I wasn't interested, and you don't ask people about their medical history. So if they say something, you listen to it, but you don't really ask them. So I didn't ask her. So she said therapy, and I accepted therapy. So I don't know, I feel like now I should know more. But she underwent some very serious therapy, and that would peel off her whole skin. And that is why she was wearing all this makeup, because it was huge peeling, because it was some kind of chemotherapy, right? So every 3 months, so the two sisters moved with her to the place where I am so that they can take care of the sister. Then one day she came to me and she said she has to go to Singapore because they have discovered some new kind of therapy, and this is once in a lifetime therapy, and she will be free of leukemia. She went there during summer, she came back. Now she did not wear any makeup. And it was a 21 years old healthy skin, right? And she's free of cancer, 100% free of cancer. So I really wish I knew what was the original therapy and what was the later therapy, but I don't know. She went back to Singapore, and so I have no idea. And I cannot write an email and ask about someone's therapy. So there are ways to get rid of a cancer, like completely. So it's not bad news. It's pretty, it is better to know what is the field and then you develop stuff around it. Yes. What time is it, Venkatesh?
4:45.
Okay, 4:45. Okay, go ahead.
One question, ma'am, you were talking about the epigenetic remarks in progression of cancer. So you were talking about methylation. So the methylation can repress as well as activates. So
Methylation in DNA is repressive.
Yeah, I'm talking about histone, histone 3, lysine 4.
It can be both.
Yeah.
But most of the time it is repressive, but it can be activating also. You're right.
And in histone 3, lysine 4 and lysine 9. So one is a.
But lysine is methylated also, that's true, but it is most often acetylated. But acetylation, usually it is a green signal, but in some cases it can be a red signal also. That's true. It depends on the whole set of modifications. Yeah.
There are certain epigenetic enzymes which LSD1, which demethylates. So it can demethylate the activate position as well as the repressor position.
Yes.
So it plays a dual role.
Yeah.
So how do we regulate this? If in a cancer condition the particular demethylase is activating, upregulated position, so it will play a dual role. So how do we manage this?
So you don't really target those guys which are playing both positive and negative roles, but there are some DNA methylases which are known, which are upregulated in cancer, and they are known to suppress tumor suppressors, not the oncogenes. So you target those guys hoping tumor suppressors will come up. So you have to make that kind of decision, which ones. You cannot target all epigenetic enzymes, but special ones. And you have to see whether it is beneficial or not. And it comes back to whether you do modeling first. You actually have to see the results to see. It is possible that they may impact other things too, but what is the trade-off if you inhibit it, right? So sometimes a little bit of damage is acceptable for the greater good, right? So you have to actually check it. And then keep in mind that small molecular inhibitors can inhibit more than one enzyme, and sometimes they can hit the right enzymes in a system. So you start with the hypothesis. Sometimes it works, sometimes it doesn't work, sometimes it works beyond your wildest dreams, and sometimes it shows exactly the opposite effect. But everything is telling you something, right?
One demethylase or methyl transferase is there enzyme, so it will transfer methylase to different position. So it will repress different genes. It can repress different genes, which one gene can promote and one gene can repress.
I totally agree with you, right? And that is why but they also have some specificity. It is not that they will do everything. So kinases phosphorylate proteins, but a kinase will phosphorylate a certain position in a protein, not all sites. So suppose a kinase phosphorylates on serine, so it is not going to phosphorylate. Suppose it phosphorylates protein A, and protein A has 10 serine sites. It's not going to phosphorylate all 10 serine sites. It depends on that kinase, which serine, and every single time it will only phosphorylate that one serine, because it depends on the tertiary structure at that site, it depends on the primary sequence around that site, so it depends on many factors. So same is true for DNA methylases also. They are not totally random that one DNA methylase will do a lot of things. There is also specificity around involved there. So when you inhibit it, you have to inhibit the right one. So that, and you have to see the trade-off. Yeah, tumor suppressors are coming up, but what about oncogenes? If you're also activating oncogenes, it's not a good target, right? So you have to determine that experimentally also. Yeah.
And we know that it is recommended to have 1.2 gram or 1.4 gram of protein per kg, like in body weight. So the people who are consuming the protein about 1.2, 1.4 gram, they are less prone to have the colorectal cancers, because I have read some articles. The person who are into fitness or bodybuilding, they are taking more protein-rich diets, and now they are developing some sort of abnormal tissue growths, and they are more susceptible to the colorectal cancer. So what's the reason behind it? Because the NPS is also working, like muscle protein synthesis there. Like if we are burning ourselves more, so our body will require more amount of protein. But still the abnormal tissue growths and protein disruptions are also observed there.
So I don't know what they are taking, right? In what amount they are taking. But one thing I know, that if they are taking protein supplements, right, like a powder, you have protein powder. So that protein powder itself may be toxic, okay? So it depends on a lot of factors. In what form? Suppose you take dal, right? So dal is healthy even if you take in large amount, but if you extract protein out of it, I don't know how much is bioavailable and how it can damage the things. So it also depends in which form you are using it. So you know, you guys have heard of Ensure drink. You have heard of it, right? So Ensure drink, so they said on their website, don't drink more than 3 times a day in the most case scenario, and usually Ensure is full of proteins, and it is also made for diabetes patients with very, very, very little sugar. So why do they say don't eat more than 3 times a day when there is no sugar? Of course there is sugar, it's bad for you. But when it is only protein rich, why not? Because excess protein what excess protein does, it upregulates nucleotide biosynthesis. If there are too many nucleotides, our body doesn't require nucleotides. So those nucleotides will make uric acid. Uric acid will just result in gout and other things. And so our body has to get rid of the excess protein also. Excess protein can create a huge problem. All these diet things came, protein-rich diet, and then they realize it's bad for you. It has to be balanced. Too much protein is also bad, right? Then this came, oh, you should eat too much fat. If you eat too much fat, body requires a lot of energy to burn fat, you will actually get thin. Then that has side effect. Then it came that, you know, you should not take sugar, but you should take sugar mimetic, like aspartame, right? Now they know aspartame is very dangerous for you, better take sugar. So it depends on what exactly they are consuming, and too much protein is really bad too. And then when you're exercising, exercise is very good, but not at the expense of damaging your muscles because you're doing so much bodybuilding. So bodybuilding is a very vast thing, right? To what extent you are building your body, what exactly is the routine, right? Exercise, you are running 30-40 minutes a day. In general, it is very good, but you have to eat healthy food. You have to supplement it with proteins. Okay, this is something you will, if you don't eat proteins, you will see in your body at your age also that you will have less energy if you don't take proteins after a while. Okay, and then once you start taking proteins, you will build it up. But supplements, these kind of monsters drink, somebody was telling me, you are going to teach, why don't you take the monster drink? Again at the airport, right? People were suggesting. I said, no, I'm not going to take monster drink. I better take a break from teaching, but not, because these monster drinks, they create a lot of problem in our body. On paper, it looks very good. If you read the content, they look wonderful, but they really mess up your system. You know, so we use lemon juice in cooking a lot, right? Sometimes the lemons are very hard and it takes a lot of time to squeeze it, right? And usually lemons in US, they are very hard, right? So it takes, they are limes and it takes a lot of, and they have very, very, very thick skin. So you squeeze and then you get some lemon juice out of it. So they have bottles. So I was really in love with the bottles, no effort, and lemon juice is there. And I used it for years and years and years. And then I came to realize, of course, it's a bottle, it has preservative. And I developed some diseases on the way. And then somebody who has no idea of science, like she's a high school person, but she was telling me a story about her sister-in-law and the preservatives. And the first thing I did is throw away the bottle. And now I buy fresh thing. And it has actually made a difference. So something as trivial as that can make a difference. So when you buy these protein powders, I don't know what is in there. I would never buy those. You know, ensure if there is a patient for a short period of time, hospital stay, that person is not able to eat, yes, ensure is good. Not for us. We better take it the proper way. So eat it in the most healthiest form, not artificial. Never go for monster drinks. Even if it says no sugar, don't go for those things. Trust me, everything counts. After spending my lifetime with diseases, the message is eat healthy, exercise, don't go for artificial things, and something I will tell you off the record. Once we are officially done for today. Okay, he is gone, so it's done, good. You are here. I thought he is gone, so this thing. Is it done for today?
If anybody has any question, they can ask 15 minutes time.
Okay, so it is still going on, right? Yeah, okay, yeah. You guys ask me. Did you guys understand what I told you, the gist of it? You will not understand everything, that's okay. I will request Professor Venkatesh to send you guys the PDF file, okay, so that you guys have it. And if you have more questions, you can obviously ask me. But if you don't have any, let's finish it off.